Okay, class, we're going to start on Chapter 4 now. And Chapter 4 is a lot more in detail about the DNA story. Just to remind you that for all the bacteria and all the archaea, DNA is the genetic material. So after we're done with this chapter, we're going to be able to describe the experiments that identify DNA as the hereditary material in cells, to discuss the evidence for the discovery of the structure of DNA, to distinguish between transcription and translation, which are going to be the next couple of chapters, and then list some examples illustrating the microbial nature of the human genome and discuss the significance of the Human Microbiome Project. Okay, so the first one to understand that there was some heredity material, it was Gregor Mendel. And Mendel was a monk, and as a monk he was able to have some time and some space to do the experiments that he wanted to do. And one of the things that Mendel's really famous for was discovering that there was something that was transmitted from parents to the offspring. Okay, and at this time Mendel only knew that there was something of a heredity factor. Okay, that's all he could really determine, was that there was something that made the offspring look like the parents, but he didn't know what that was. It would be years before we would know that that would be DNA. Okay. Now, one of the first people to put some of this together a bit more, to start using microbiology, okay, that's Frederick Griffin. Harmful the harmful
started
DNA
working with
from the strain that caused death was actually moving into the strain. Oswald Avery and Hershey and Chase used E. coli
one
and
sure way to win a
Nobel Prize is to find the structure of DNA.
We need to remember,
and Watson and Crick, a double helix is made up of nucleotides. Those nucleotides are adenine, are the things that are in the center of the guanine, and cytosine, blue here, or AT. And this is a very important rule here, that there are kind of two ways is equal to the number of T's that you could have DNA replication. So gene expression. We have our original strand of DNA here. So gene expression is how we go from the DNA and to the protein. Like I said, gene expression is how we get a protein from our DNA. In transcription, we use something called the RNA code that translates this. Okay, so you have your piece of DNA here. So in the positive transcription, okay, the mRNA. And so here you can see that you can have an incoming, and here's again just a summary that translated. We have a DNA
Okay, the last part of our chapter is on genes and chromosomes, human and microbial sources. Okay, so you may know that recently the Human Genome Project was finished, and there are about 3 billion genes in a person. Okay, sorry, 3 billion nucleotides in a person. Okay, as opposed to a bacterial sequence, which is only about 4.7 bases and only 4,300 genes. So obviously, bacteria are much more streamlined than a human. We don't have as much so-called junk DNA, but everything in a bacterial genome is probably a little bit more important because there is nothing that's junk. Okay. And so one of the reasons why we spent all the money to sequence the Human Genome Project, it was that it would be used for identifying defective genes. Okay, so a number of genes that can be used or implicate in disease can be found that way. And also new medicines can be discovered to be able to make use of genes that currently aren't being used to develop new pharmaceuticals. Okay, so hopefully we will be able to put all that sequencing work to good use and find new drugs. Okay. Now, one of the things, like I mentioned when I was talking about the genetic code, is we all have these same genetic code. It doesn't matter what the organism is, we all have the same genetic code. Okay, so in addition, we have a number of genes that are similar to those of the microbes. Okay, there are about 200 genes found in bacteria and about 6,000 genes found in yeast, and these things are passed on just like, you know, sort of normal genetics, and where we start getting into a little bit of gray area is that there are a number of human genes that look like viral genes. Okay, and many of those are things that are reverse transcriptase genes. Okay, so that is they're a virus that's made out of RNA, and then when it infects into the human, it will reverse itself and make itself into DNA. And so just like HIV does. And those little things are little markers that get put into the human DNA, and we can and we propagate those viruses. Okay. And it's kind of interesting. So I told you we have a human done, and that's relatively new. It was in the 90s that the human genome was sequenced. But the number of bacteria, because they're so much smaller, again, in the order of millions of base pairs as opposed to billions of base pairs, we can do a whole lot of bacteria. And I haven't looked at the data in a couple of years, but we were up over 500 bacteria that have been completely sequenced. Okay, and so that's just one of the ways that we can do that comparative sequencing thing. Okay. One of the things that we want to do with all that data is to be able to do microbial genomics, so we can analyze and compare different genomes. Okay, and so one of the things you could think about with that is that we could say make safer food products, or we can find ways that bacteria infect us. So it made me think of some very malicious bacteria that really wants to infect you, even though we said in the first chapter that causing disease is the minority player of the bacteria. But if you do get a bacterial infection, if you know all the genes, you might be able to develop a pharmaceutical that could selectively target that that bacteria and hopefully kill it in a person. Okay. And like I said, we can compare different genomes, which is a process called metagenomic, and of all the things that have been sequenced, 67% have been bacterial. Okay, and again, we can, because there are so few nucleotides actually sequenced, you can actually get quite a bang for your buck by sequencing bacterial genomes. One of these days, I hope to be able to sequence my own. Now, I've said before that a microbiome is all the bacteria that live in a certain place, and this is a slide showing the different bacteria and where they mostly live. Okay, and you can look at the chart, and I mean, the vast majority of the things on the human skin are Staphylococcus epidermidis. Okay, but there are certain places on the body that have certain types of bacteria that are always present there. Okay, and that's what we're seeing in this slide. You can go through and look at each of the categories on there, but you can see which bacteria are pretty much always there, and so they make up the skin microbiome. Okay, and usually what these are doing is improving health and improving the immune system. Okay, so what we should have learned, that DNA is blueprint for all living organisms, that how you express those genes is how we get the organism to actually work, and that microbial and viral contribute to the human genome. Okay, like I said, there are a number of bacterial genes and viral genes that have incorporated into the human beings. Okay, and a thought question: how would our lives be different if we had no human microbiome? Okay, my short answer to that is you would get a lot more diseases. Okay, on a good note, you wouldn't smell. Okay, you could go out and run around and sweat as much as you wanted to, but there would be no bacteria to cause the odor. Okay, so those are just two examples. Please think of some other ones, and we can maybe talk about those.